Hi everybody, so today we're going to be talking about the resting membrane potential of a nerve, and this is looking at a nerve right here, and if you recall right here, these are your dendrites, this is your cell body, this part here is called the axon hillock, that's the axon hillock, and then this is my axon, and then at the end we have the axon knobs and terminals. So let's take a look at this. What I'm going to do is I am just going to take this area that is right here and I'm going to make this larger. I'm going to blow this up. So let's erase all this. Again, all I'm doing is making this area larger. So this is what my membrane is going to look like, okay? If I just made that part larger, I'm going to go like this. All right, and then let's go like this. Over here again would be my hillock, and this would be my cell body, and my dendrites would all be over in here. But again, I'm only interested in this part over here. So first, when we talk about a cell, we say that the cell is polarized. What do we mean by polarized? What we mean by polarized is there's a difference in charges that are separated by the membrane. So on the outside, we have more positive charges, and on the inside, it's more of a negative charge. This is what we call polarized, okay? Now, the difference in these charges is called the potential. So this is my membrane. Because the difference in the charges is across the membrane, we call it the membrane potential. It's the difference in the charges. So let's take a look at this real fast. And on the inside of your cell, of your neuron, you have negatively charged proteins. You also have negatively charged amino acids, but I'm not going to write those right now. You have negatively charged proteins, okay? So there's my protein. They provide most of the negative charge on the inside of my neuron, okay? So like we said, the inside is negatively charged, the outside is more positively charged, and we'll tell you why in just a minute, but we want to talk about the potential real quick. So here's the thing. Let's say that this protein, okay, this is not exactly how it works, I'm just giving you an idea. Let's say this protein has a charge of negative 100, but outside we have a charge of positive 30. Well, the difference between those, negative 100 plus positive 30, would give me an inside resting membrane potential of negative 70. Now, some books will be a little bit different. I've seen this as low as negative 60. I think I've even seen once where it said negative 80. We're just going to say negative 70 because that's pretty much the average of what this is. So we have negative 70. That's my resting membrane potential. Now let's talk about what's going on here. I'm going to erase this because I just want you to remember this is negatively charged. I'm going to erase this. Just remember the outside is mainly positive. Inside of my neuron, like we said, we have the negative charges, but inside my neuron, we have potassium ions, okay? I just drew a cluster right here. This goes throughout the whole neuron, okay? But I just drew it right here just to make it a little easier for what we're explaining. So on a resting nerve, we have more potassium ions inside than outside, than out here. So there's more of these inside than outside. On the outside, we have sodium ions. Okay, so this is what we have on the outside. We have sodium ions on the outside. So now we have more sodium ions outside than inside, more potassium ions inside than outside. I know it sounds confusing, but if you just look at the picture, I have more out here than in here, and more of this in here than out here. When the membrane's at rest, what's going to happen, well, before we get there, in the membrane, there is something called leak channels. So this is a potassium leak channel. All right, that's my potassium leak channel. And I'm just going to finish drawing my membrane here. All right, there's my potassium leak channel. And then we have our sodium leak channel. So here's my sodium leak channel. Sodium leak channel. Okay, and then between these two, you're going to find something called the sodium potassium pump. So let's draw this here. When the membrane, when the neuron's at rest, what's going to happen is potassium is going to leak out through the leak channel. So here goes my potassium through the leak channel. Okay? And there's a reason why. The reason why is, once again, simple diffusion. If you notice, on the inside, I have more potassium ions than I have outside. So because there's more, this is going to leak out. Okay? Now, I also have sodium leaking in. So here comes the sodium, here comes the sodium, here comes the sodium. All right, so now I get sodium leaking in. So now the reason sodium leaks in is because I have more sodium ions outside than inside. So because of the fusion, it's going to leak in. Plus, remember, this is positively charged and inside's negative. So if it's coming in because of it's the positive charge, and this is negative, we call that an electrical gradient. Okay, it's electrical because we have positive and negative charges being attracted to each other. If it comes in because of the fusion and being more ions out here, we call that a chemical gradient. Okay, so there's an electrical gradient and a chemical gradient. Electrical gradient has to do with the difference in charges. Chemical gradient has to do with the difference in the number of ions. Okay? So now, if you notice on here, what happened is I drew 2 potassiums, but I drew 3 sodiums. So for every 2 potassiums, I'm going to write this over here. For every 2 potassiums that leak out, 3 sodiums leak in. Okay, 3 sodiums leak in. Okay, so we got 1, 2, 3, 1, 2. Here's what happens here now, is because these leak out, remember we said out here is positively charged, and this is positively charged. It does not want to be where it's positively charged, so what happens is my sodium potassium pump is going to leak, or pump, it's actually going to pump these back in. This is active transport, because now we're going from an area of low concentration of potassium to high. And as you probably learned in biology, whenever I have active transport, I am going to use energy. So I'm going to go from ATP to ADP. Okay? The other thing that happens now is my sodium is pumped out as fast as it comes in. And once again, this is going to require energy. Okay? So for every 2 that go out, 3 come in. What that will do is that will maintain this negative 70 millivolts, that balance. Okay? So that's my resting membrane potential. So let's review real quick. We said outside. First of all, it's polarized because of the fact that outside you have more, you have a difference in charge between the outside and inside, right? So it's polarized, and it's separated by a membrane, cell membrane. So that's why they call it polarized. Then we have a potential difference, right? The difference in those charges. In this case, we said it was negative 70. We said there's more sodium inside than outside. More potassium inside than outside. More sodium outside than inside. For every 2 sodium that go out, for every 2 potassium that go out, 3 sodium come in. So just to make this clear, the K is potassium, okay? And Na is sodium, just because I've mixed up the words a few times there. So for every 2 potassium that go out, 3 sodium come in. And then it just keeps going on and on. Now, this is not only right here. Can go throughout the whole thing. You're looking at this as a 2-dimensional object. It's actually 3-dimensional, so imagine this being anywhere on the tube that we call the axon, the hillock, or even over here, the membrane. Okay? And that's it. That's our graded potentials.